Scientific Critique and Falsification of the RNA World Hypothesis for Abiogenesis. Summary. In this video, we present a scientific critique of the RNA World Hypothesis for Abiogenesis, based on the scientific literature. The RNA World Hypothesis is the best conjecture available for the origin of life. However, it has been falsified, or defeated, by the scientific evidence. The origin of life remains an intractable problem, from a naturalistic perspective, just as we would predict from an intelligent design perspective. Abiogenesis experiments confirm that intelligent design is required. In intelligent design of the experiments, intelligent selection of precursor chemicals, intelligent purification of the precursor chemicals, intelligent removal of conflicting reaction products that stop the reactions of interest, for any progress towards life-relevant systems. All of this points to the fact that intelligent design is required for the origin of life. In the rest of this video, we present a scientific critique of the RNA World Hypothesis for Abiogenesis, based on the scientific literature. Scientific references are listed in the description, or pinned comment, of this video. Introduction. The RNA World Hypothesis proposes that early life was based on RNA molecules acting both as genetic material and as catalysts, before the evolution of DNA and proteins. While this idea is widely discussed, it faces numerous scientific critiques and falsifiers, or defeaters. Scientific researchers have pointed out several falsifiers that an RNA world origin of life must overcome. Quote, common criticisms are that RNA is too complex to emerge de novo in a prebiotic environment, that catalysis is a relatively rare property of RNA and requires implausibly long strands, that the catalytic repertoire of RNA is too limited and that it is difficult to envisage scenarios in which precursors and feedstocks occurred at sufficient concentrations to allow replication and evolution. References 5 and 11. Below is an overview of the main critiques, followed by in-depth discussion of specific issues. Chemical instability of RNA in prebiotic conditions. RNA and its building blocks are not chemically robust in early Earth environments, raising defeaters related to their non-persistence. Difficulty of prebiotic RNA synthesis, forming the components of RNA, bases, ribose sugar, and phosphate, under primordial conditions and assembling them into polymers is empirically impossible without intelligent design intervention by experimenters. Challenges in RNA replication and catalysis, even if RNA could form, achieving self-replication and catalytic function, comparable to a primitive metabolism, without proteins is empirically impossible. Each of these points is elaborated below, with references to recent studies and peer-reviewed research that highlight the key findings and counterarguments by critics of the RNA World Hypothesis. Chemical instability of RNA in prebiotic conditions. One fundamental defeater is that RNA is chemically too unstable to survive in the harsh conditions of the early Earth. RNA's ribose phosphate backbone and nucleotide bases are prone to decomposition. For example, the four canonical RNA bases, adenine, guanine, cytosine, uracil, break down rapidly at high temperatures. A classic study by Levy and Miller, 1998, measured base stability and found that at 100 C, a temperature relevant to hot hydrothermal environments, adenine and guanine have a half-life of approximately 1 year, uracil approximately 12 years, and cytosine only 19 days, reference 1. The stability of the RNA bases. Implications for the origin of life. PubMed. In other words, RNA's bases would decompose far too quickly in a boiling hot, primordial soup, for an RNA-based life to get started. Unless life arose very fast, a high-temperature origin could not rely on the standard nucleobases, reference 1. Even at 0 C, cold early Earth scenarios, cytosine is unstable, half-life approximately 17,000 years, indicating that the GC base pair would not have been viable unless life emerged very quickly, reference 1. Beyond the bases themselves, the RNA polymer is also vulnerable. RNA is often called a prebiotic chemist's nightmare, due to its intrinsic instability. It's a large polymer made of sugar-based subunits and has bonds, like the phosphodiester link, that are thermodynamically prone to hydrolysis, breakdown, in water, reference 2, asphalt, water, and the prebiotic synthesis of ribose, ribonucleosides, and RNA. PubMed. Water can actually be an enemy to RNA. This is dubbed the water problem. Benner and colleagues note that many bonds in RNA will spontaneously hydrolyze in aqueous solution, meaning RNA would fall apart unless conditions were just right. Reference 2. For instance, the 2' OH group on ribose can attack the backbone, causing the strand to cleave. In a continuously wet environment, long RNA chains would not persist for long. This instability is a major issue given that the prebiotic Earth was covered in water. Moreover, energetic events common on the early Earth would threaten RNA. UV radiation from the sun breaks chemical bonds, and without an ozone layer early on, any surface RNA would be quickly degraded. Periodic events like meteor impacts or volcanic activity would also reset the chemical environment. Researchers have pointed out that a large asteroid impact would boil off oceans and destroy complex molecules, meaning any RNA would have to reform afterwards. Reference 1. Fine-tuned conditions. Experiments confirm the need for extremely fine-tuned experimental conditions for any progress towards life relevance. The requirement for such finely tuned conditions underscores the critics' point that RNA is not very hardy on its own. In summary, scientific critics argue that RNA would rapidly degrade under realistic prebiotic conditions, especially in heat or sustained water exposure, making it empirically impossible as a starting point for life without some form of shielding or extremely favorable conditions. Reference 2. This points to intelligent design needed for abiogenesis. Empirical impossibility of prebiotic synthesis of RNA components. Even if RNA is inherently unstable, one could still argue life got lucky if there was an abundant supply of RNA building blocks that somehow managed to polymerize. However, another major critique is the lack of any demonstrated empirically possible prebiotic route to make the components of RNA in sufficient quantity and purity. The RNA World Hypothesis assumes that the early Earth had a supply of the 4 ribonucleotides, adenosine, guanosine, cytidine, uridine or their analogues. But forming these is extremely chemically complex, requiring intelligent design intervention by experimenters. Ribose sugar falsifier. Ribose is a 5-carbon sugar required for RNA. The classic idea for its prebiotic formation is the Formose reaction, which produces sugars from formaldehyde. But this reaction produces a complex mixture of sugars, not predominantly ribose. Worse, if there are nitrogenous compounds, like amino acids, peptides, or bases, in the same soup, they react with formaldehyde or the sugars to form tarry substances, effectively quenching the formation of ribose. Reference 3. The RNA world, a critique. This is often referred to as the asphalt problem. Without careful intelligent design control by scientists, organic reactions lead to complex, unhelpful gunk. Reference 2. Stanley Miller, famous for the Miller-Urey experiment, and colleagues showed that ribose itself is very short-lived in solution. At neutral pH, it decomposes surprisingly fast, on the order of days or weeks. Reference 3. They concluded it's very unlikely that sugars were available as prebiotic reagents in any significant concentration. Reference 3. Base, nucleobase, synthesis falsifier. The RNA bases, purines and pyrimidines, are also very difficult to make under primordial conditions. Adenine can be made from hydrogen cyanide, HCN, in the lab, but the yields are extremely small, on the order of 0.1% or less, and require concentrated HCN and ammonia in strongly alkaline conditions. Reference 3. Such conditions are incompatible with the conditions needed for ribose. For instance, you can't form sugars in the same hot alkaline cyanide-rich soup that makes purines. Reference 3. So the base and sugar production pathways conflict with each other. Moreover, even if some adenine did form, it wouldn't stick around. Adenine reacts further, it can deaminate or undergo ring opening, with a measured half-life of only approximately 80 years at 37 C in water. Reference 3. Cytosine has an even bigger problem. It hydrolyzes to uracil on similar timescales. In short, the necessary bases would not accumulate in the open ocean or lakes, but instead would break down or react with other chemicals. Reference 3. Phosphate falsifier. To build nucleotides, a source of phosphate is needed, for the backbone linkage. Most phosphate on early Earth was locked in insoluble minerals like apatite. In lab syntheses, Researchers use unrealistically high concentrations of free phosphate or activating agents, but in nature phosphate is sparingly available. This phosphate problem notes that common phosphate minerals don't dissolve easily, so getting enough phosphate in solution to make nucleotides is empirically impossible in a life-relevant early Earth environment. Reference 4. Prebiotic chemistry. A review of nucleoside phosphorylation and polymerization (PMC). Assembly of the nucleotide. Even if one had ribose, a base, and phosphate, joining them in the correct way, beta-D nucleoside linkage, and then 5' phosphate, without enzymes is non-trivial, i.e., extremely unlikely without enzymes. There are many possible isomers and wrong attachments that could occur. The specific combination needed for RNA is just one among a multitude of outcomes if you simply mix the ingredients. Orgel and others have pointed out that getting a high yield of just the 4 correct nucleotides, and not a mess of similar molecules, would be extremely low probability on the primitive Earth. Reference 3. Given these hurdles, Gerald Joyce and Leslie Orgel once dryly remarked that the idea of an RNA replicator emerging de novo on the primitive Earth has to be akin to a miracle. It requires a series of fortunate chemical events that we have trouble recreating even in the lab with purified chemicals, with intelligent design intervention by intelligent experimenters. Or as Stephen Benner put it, RNA is a nightmare for prebiotic chemists because it requires a combination of building blocks that are very difficult to make and would fall apart without carefully controlled conditions. Reference 2. This points to intelligent design required for abiogenesis. No experiment has demonstrated an empirically possible prebiotic synthesis of RNA from start to finish. The experiments require intelligent design interventions at each stage. As Benner et al., 2012, note, chemists can sketch out a discontinuous, multi-step pathway in which early Earth processes produce formaldehyde and HCN, then sugars, then bases, then nucleosides, then nucleotides, and finally RNA. Reference 2. However, no experiments have joined together those steps without human intervention, and many steps only work in intelligently designed idealized scenarios, adding reactants in a specific sequence, purifying intermediates, etc. Reference 2. If any step is done in a one-pot mix, it yields a tarry sludge of side reactions, the asphalt problem. Reference 2. Some bonds needed for RNA are practically impossible to form under any realistic early Earth conditions. Reference 2. For example, the direct attachment of the base to ribose is impossible under realistic early Earth conditions. In summary, the synthesis problem is a major critique. Scientific critics show that the spontaneous assembly of RNA's building blocks on prebiotic Earth was extraordinarily improbable. Even in 2023, a review concluded that how nucleosides were phosphorylated and polymerized is still perplexing, noting the phosphate availability issue and the need to address chirality and many other factors. Reference 4. This skepticism has led many to consider that maybe RNA wasn't the first polymer at all, or that it needed significant help. My comment: abiogenesis requires intelligent design. Defeaters in RNA replication and catalysis. Even if we grant that some RNA could form, another critique asks, how would it replicate and evolve? For the RNA world to get going, there must have been a self-copying RNA, a replicase ribozyme, or at least a set of RNAs that could catalyze each other's formation. This is a stringent requirement. Critics point out several issues here. No self-replicating RNA known. Scientists have not been able to demonstrate a robust, continuous RNA self-replication in the lab without protein enzymes. We have examples of partial progress. For instance, a 2016 study evolved an RNA that can copy RNAs up to 206 nucleotides, but it still required a stable supply of activated nucleotide triphosphates and worked only under specific conditions. And the RNA could not copy itself fully, just other sequences. Reference 5. The difficult case of an RNA-only origin of life (PMC). A true RNA replicator, one that could arise spontaneously and indefinitely make new copies of itself, remains empirically impossible. As a 2019 review summed up, it has not yet been possible to demonstrate robust and continuous RNA self-replication from a realistic feedstock, i.e., activated monomers, highlighting that this core prediction of the RNA world is still defeated or falsified. Reference 5. Template inhibition. Strand reannealing. One technical but critical problem is that when an RNA strand copies another, you get a double-stranded RNA duplex. The complement binds to the template. In modern biology, helicase enzymes separate strands. Prebiotically, how would strands come apart? The issue is that RNA duplexes are very stable. Experiments show that if you melt strands apart, by heat, and let them cool, they zip back together much faster than any copying reaction can make new strands, reference 5. This means an RNA replicase would quickly find itself stuck. Its newly made complement binds back to it, making a double helix that can't be used further unless something, like a temperature cycle or a clay surface, helps pull them apart. This strand separation problem is a big hurdle for continuous replication, reference 5. Intelligent workarounds require very specific combinations of conditions that would be impossible to come by consistently in nature, reference 5. Replication fidelity. Error threshold. Early replication would be error-prone. There were no polymerase enzymes to ensure accuracy. Manfred Eigen in 1971 formulated the concept of an error threshold for early genomes. If the sequence is too long and copying is too inaccurate, information will be lost faster than it's gained, preventing Darwinian evolution. Calculations indicate that a replicator with typical RNA error rates could only be very short. For example, an RNA that copies itself with 90% fidelity, which is actually high for a non-enzymatic process, can only be about 12 nucleotides long, before it mutates away its information, reference 6. Eigen, M. 1971. Reference 7. The Origins of the RNA World. At 70% fidelity, it could only be a few nucleotides long, reference 6, 7. These lengths are far too short to encode any meaningful catalytic function. By comparison, even the smallest ribozymes known, like a self-cleaving ribozyme, are approximately 30 to 40 nucleotides, and a functional replicase needs more than 100 nucleotides. This creates a paradox. To have a faithful replicator, you need a long sequence to fold and catalyze. But to maintain a long sequence, you need faithful replication. It's a chicken and egg problem often called Eigen's paradox. Critics argue this makes the spontaneous emergence of an RNA replicase vanishingly unlikely, i.e., empirically impossible, reference 6, 7. Limited catalytic repertoire. RNA can catalyze some reactions, phosphate transfer, peptide bond formation in the ribosome, etc. But its abilities are limited compared to proteins. The RNA world requires that crucial metabolic reactions, like nucleotide activation, membrane formation, energy handling, were catalyzed by ribozymes. While ribozymes have been discovered or engineered for some reactions, many essential activities have no ribozymes. For instance, no ribozyme can perform robust template-directed polymerization of RNA from monomers better than very slowly, too slowly for life's requirements. The best RNA polymerase ribozymes still struggle with copying sequences approaching their own size, and they work only on specific templates or require helper oligonucleotides, which are provided by intelligent design by the experimenter. Joyce and Orgel, reference 8, noted that it seems extremely unlikely that a pair of complementary RNAs existed where each could help assemble the other from free nucleotides, given the inherent difficulties of template copying. Monomers fall off, side reactions occur, etc. To them, certain obstacles seem intrinsic to the template-directed condensation of activated mononucleotides in water. In simpler terms, naked RNA is not up to the task of starting an evolutionary process by itself. Requirement for supporting systems. An often overlooked point is that even an RNA replicator would need a steady source of activated nucleotides, chemically charged monomers, to keep replicating. In modern labs, intelligent and purposeful researchers feed experimental RNA replicators with chemically prepared nucleoside triphosphates or similar. Prebiotically, what regenerated the supply of activated building blocks? If the environment only provided raw nucleotides occasionally, an RNA replicator would quickly run out of fuel. It is empirically impossible to have an ongoing, ready supply of substrate without any proteins around to synthesize them. Abiogenesis experiments confirm the need for intelligent design interventions by experimenters. In summary, The replication and catalysis critique essentially says, even if we had some RNA, why should it spontaneously form a self-replicating, evolving system? It seems chemically and kinetically implausible, i.e., empirically impossible in practice. Despite decades of work, scientists have not produced any Earth-like scenario where RNA can emerge and sustain Darwinian evolution on its own. Critics take this as evidence that the original RNA world idea does not work. It might require either extraordinary luck or some missing piece, e.g., intelligent design, to work. This naturally opens the door to alternative hypotheses for the origin of life, such as intelligent design. Defeater in inventing the genetic code. Another critique comes from the empirical impossibility of inventing the genetic code. The genetic code is the set of rules by which RNA sequences, mRNA codons, are translated into proteins. It's empirically impossible to see how a complex translation system, ribosome, tRNAs, aminoacyl tRNA synthetases, etc., could evolve if you start from an all-RNA world and then somehow proteins appear later. Scientific critics like Charles Carter and Peter Wills have argued that there are insuperable problems with the code emerging in a purely RNA context. Reference 10:5. Conclusion. The RNA World Hypothesis has been useful as a framework for origin of life research, but it has been falsified by the scientific evidence. The scientific critiques summarized above highlight how chemically demanding, i.e., empirically impossible, it is to go from prebiotic ingredients to an RNA-based proto-life. RNA is fragile in water and heat. Reference 2. Empirically impossible to make without enzymes or chemists. Reference 9. And empirically impossible to get it to copy itself without a lot of pre-existing cellular machinery. Reference 5. The origin of life remains an intractable problem, from a naturalistic perspective, just as we would predict from an intelligent design perspective. Abiogenesis experiments confirm that intelligent design is required. In intelligent design of the experiments, intelligent selection of precursor chemicals, intelligent purification of the precursor chemicals, intelligent removal of conflicting reaction products that stop the reactions of interest, for any progress towards life-relevant systems. The scientific references are listed in the description, or pinned comment, of this video. Thanks for watching. Please like, share and subscribe, and comment below if you agree, or would like to add your thoughts. If you would like me to respond to your comment, please make a level 1 comment, directly below the video. I will respond to all polite level 1 comments even if it takes me a while, due to other commitments.